السلام عليكم أعزائي طلبة المرحلة الثالثة. هذه المحاضرة الأولى للفصل الثاني من مادة Medical Parasitology. At the end of this lecture, the student able to define the protozoa as a group, list the classes of protozoa, describe the main characteristic features of protozoa, describe the first phylum Sarcomastigophora of protozoa, then describe the first member of protozoa, Entamoeba histolytica. We all remember this classification of parasites. The parasites either protozoa, unicellular parasite, or metazoa, multicellular parasites. The both belong to kingdom Animalia. Helminthes, as previously classified to nematode, cystode, trematode. While the protozoa we will classify in amoebae, flagellates, sporozoa, ciliate. What are protozoa? Proto mean first, zoa animal. So protozoa mean first animal. الأوالي. It is single-celled eukaryote organisms. The cell unit performs all of the necessary function of the life. إذن هنا خلية واحدة تقوم بكل وظائف اللي تحتاجها الأورجانيزم للحياة. Differences between protozoa and metazoa in morphology, as we said previously, protozoa it is unicellular or single cell unit, while metazoa multicellular number of cells making up a complex individual. Physiology, these single cells perform all the function for life, like reproduction, digestion, respiration, excretion, etc. While metazoa, each special cell performs particular function. Example, amoebae for protozoa and tapeworm for metazoa. The taxonomic classification of protozoa. Protozoa, it is subkingdom of kingdom Animalia. Contain 4 phyla, Sarcomastigophora, Ebicomplexa, Ciliophora, and Microspora. Sarcomastigophora divided to 2 subphylum. Sarcodyne contain amoebae, and Mastigophora contain flagellate. Ebicomplexa, example Plasmodium. Ciliophora, the parasitic organism Balantidium coli. Microspora, example Enterocytozoa. Morphology and biology for the protozoa. Shape. There is no one shape or morphology which include the majority of protozoa. The shape range from amorphous or ever-changing forms. This will see it in amoebae, or rigid forms. These rigid forms may be spherical or regular ovoid. Some have bilateral symmetry and other have torsion along the longitudinal axis. As we see in these 3 pictures, first one for amoebae, second for trichomonas, and the spindle shape for Leishmania bromastigote. Internal features. All protozoa have certain morphologic features like nucleus, cytoplasm, which may be divided in some to endoplasm and ectoplasm. For the nucleus, contain chromosomes that regulate the growth and reproduction and determine the genetic characteristic of species. In amoebae, there is also in the nucleus karyosome that's situated in a relatively viscous nucleoplasm. The nucleoplasm is bounded by this nut membrane that has many spores connecting to the cytoplasm. The cytoplasm can be differentiated in some protozoa to endoplasm and ectoplasm. Endoplasm surrounding the nucleus consists of dense, finely granule protoplasm that function in the digestion of ingested food and other process, while the ectoplasm serve for locomotion للحركة, for obtaining and ingesting food, and for respiration and excretion. These processes are tightly controlled by the plasma membrane, maintaining normal concentration of plasma. Food reserves in the form of glycogen mass may be present in the cytoplasm. Feeding protozoa may be absorb food via their cell membranes. In amoeba and other intestinal forms surround food and engulf it into food vacuoles which present in the endoplasm. Others protozoa, like Palletidium coli from the ciliate, have opening, we call it mouth pores, or cytostome, in which they sweep food into food vacuoles and contractile vacuoles. All protozoa digest their food in stomach-like compartments called vacuoles. Protozoa motility, as we divided to subgroups, flagellate motile by flagella, ciliate motile by cilia, amoeba moved by pseudopodia, sporozoa gliding motility or have epicomplexian compartment, as we studied in this group. Protozoa reproduction. Some protozoa reproduce asexually by binary fission or multiple fission or endogeny, or maybe reproduce sexually by gametogamy, fusion of gametes, or conjugation, al-iqtiran. Some protozoa use combination of sexual and asexual reproduction, as we will see it in coccidia. This picture indicating the asexual reproduction. We divided to binary fission. It is the method of reproduction in which a single parasite divided either longitudinally or transversely into two parasites. Mitotic division of a nucleus is followed by division of the cytoplasm. In amoeba, division occurs along any plane. But in flagellate, division is along longitudinal axis and in ciliate in the transverse plane, as we see in this picture. The second asexual reproduction, it is multiple fission or schizogony. This occurs in a plasmodium, exhibits schizogony in which nucleus undergoes several successive division within the schizone to produce large number of merozoites. Endogeny, some protozoa like toxoplasma multiply by internal budding, resulting in the formation of two daughter cells. While sexual reproduction, we divide it to conjugation and to gametogamy. Conjugation, it occurs in ciliate. It is conjugate two organisms joined together and exchange nuclear material, then divided into two organisms. While gametogamy, it occurs in sporozoa or coccidia. It is united by male and female gametes to produce zygote, which gives rise to numerous sporozoites or sporogons. This picture for ciliate, which reproduct asexually transfer binary fission and sexually by conjugation. Life cycle. Some protozoa have simple life cycle because it is have trophozoite stage only and single host binary fission, asexual reproduction like Entamoeba gingivalis, which present in disease gum and teeth, and Trichomonas like Trichomonas tenax and Trichomonas hominis. Trichomonas vaginalis and Entamoeba fragilis, all these parasites have trophozoite stage only and reproduce sexually by binary fission. So it have simple life cycle. Parasites have complex life cycle with multiple stages and proliferative stages. May have trophozoite and dormant stage, cyst stage. Trophozoites, it is the nourish, actively feed, and grow stage, while the cyst stage can survive in harsh condition outside of the host because it have hyaline wall, allows them to transfer transmission from one host to another. Like Entamoeba histolytica have two stages, trophozoite and cyst stage. Entamoeba harshmani the same, Entamoeba coli, Entamoeba xnane, and Amoebiasis buccioli. In some protozoa, like Leishmania, Trypanosoma, and Plasmodium species, they have two successive hosts are required in their life cycle, in which one of them it's blood-sucking insect like mosquito or sandfly. It is the vector, and it is essential for the life cycle because it's have opportunity for multiplication of progeny. We will start with intestinal protozoa. As we say before, it is unicellular eukaryote organism. It is have typical or fecal-oral life cycle. Some of them have trophozoite stage only, others have trophozoite and cyst stage. The trophozoites, it's the feeding, motile, and replication stage. After encystment, will develop to cyst stage, contain hyaline wall. It is based in feces and resistance to harsh environmental condition and infective to the second host. Then by excitation, transfer to trophozoites. This life cycle, we will see it in flagellate like Giardia lambelia, Entamoeba fragilis, Chilomastic minili, Trichomonas hominis, and Trichomonas tenax. In amoebae like Entamoeba histolytica, Entamoeba dispar, Entamoeba coli, Entamoeba harshmani, Entamoeba xnane, Amoebiasis buccioli, and also in epicomplex like Cryptosporidium species and Isosporabili, and in ciliate like Bacillium coli. Some of the trophozoites will develop into cysts in a process we call it encystation or encystment, in which the nucleus multiplies by binary fission instead of undergoing replication. So encystation, it is a process for protection because it is produce hyaline membrane around the trophozoite, and for multiplication because the nucleus in some protozoa will multiply in the cyst. The cysts are characterized by resistant wall and are excreted with the feces. We will start with the group Amoebae. Amoebae are unicellular organisms belong to the phylum Sarcomastigophora, subphylum Sarcodyne. It is common in the environment. Many of parasites of vertebrate and invertebrate belong to this subphylum. A few species inhabit the human intestine, and only Entamoeba histolytica it is pathogenic intestinal amoeba for the human. If we classified the amoeba, we have intestinal amoeba like Entamoeba histolytica, Entamoeba dispar, Entamoeba coli, Entamoeba boliki, Entamoeba hartmanni, Entamoeba gingivalis, Entamoeba xnana, Entamoeba buccillae. All these, it is intestinal amoeba. Only Entamoeba histolytica, it is pathogenic. The others, it is non-pathogenic. And we have free-living amoeba like Naegleria and Entamoeba species. They are free-living, but it is when produced to the human, it is opportunistic pathogen and will produce disease and serious disease for the human. We will start with Entamoeba histolytica, the disease caused amoebiasis or amoebic dysentery or bloody diarrhea. Entamoeba histolytica were recognized as pathogenic amoeba associated with intestinal and extra-intestinal infection. The other species are important because they may be confused with Entamoeba histolytica in diagnostic investigation and indicating the filth contamination with filth material. So we have two causes to study other amoeba. Instead, they are non-pathogenic. It is the Entamoeba histolytica. It is typical fecal-oral life cycle, and the habitat it is large intestine of a human. Morphology, Entamoeba histolytica have four forms or stages: trophozoite, pre-cyst, cyst, and metacyst stage. Start with trophozoite. Trophozoite, it is vegetative or growing stage of the parasite. It is only stage present in tissue. It is irregular in shape, and the size range from 12 to 16 micron. The average is 20 micron. It is large and actively motile in freshly passed dysenteric stool, while smaller in convalescent and carriers. Contains cytoplasm, which differentiate to ectoplasm is clear and transparent. Inner endoplasm is granular. The endoplasm contain nucleus, food vacuole, erythrocyte, RBCs. It is the characteristic features for the Entamoeba histolytica and differentiate it from other amoeba by presence of erythrocytes, RBCs in their vacuoles. Also presents leukocytes and tissue deposits. The trophozoite from acute dysenteric stool often contain phagocytoid erythrocytes, as we said before. This feature is diagnostic as phagocytoid red cells are not found in any other commensal intestinal amoeba. هذه ملاحظة مهمة حتى نفرق الأميبا Entamoeba histolytica عن باقي الأميبات. Pseudopodia in trophozoite of Entamoeba histolytica is finger-like projection formed by a sudden jerky movement of ectoplasm in one direction, followed by the streaming in of the whole endoplasm. Pseudopodia formation and motility are inhibited at low temperatures. A nucleus is spherical and contains central karyosome. The nuclear membrane is lined by a rim of chromatin distributed evenly as small granules. The trophozoite divided by binary fission in every 8 hours. Trophozoites survive up to 5 hours at 37 degrees and are killed by drying, heating, and chemical sterilization. Therefore, the infection is not transmitted by trophozoite. Even if live trophozoites from freshly passed in stool are ingested, they are rapidly destroyed in stomach and cannot initiate infection. هاي ملاحظة مهمة إنه حتى لو ابتلع التروفوزويت وهو alive لا يمكن أن يسبب إصابة بسبب الأحماض والإنزيمز الموجودة بالستومك وهو very sensitive to these chemical components. This is pictures for the trophozoites of Entamoeba histolytica. The nucleus characteristic features contain central karyosome, and the inner nuclear membrane contain regular chromatin dot. This is electron microscope for Entamoeba histolytica trophozoite. The second stage of Entamoeba histolytica is pre-cyst stage. It is round or ovoid, sluggish movement, a blunt pseudopodia, no RBCs. In vacuole, the size range between 10 to 12 micron. The picture B, it is represents the pre-cyst stage. The third stage is it is cyst, found in the lumen of large intestine and never found in tissue. It starts as uninucleated, binucleated, and quadrinucleated. Only quadrinucleated cyst, it is the mature cyst, and it is effective for the second host, while the other, it is immature cyst. Quadrinucleated cyst, as we see, is the infective stage. Multiply by binary fission. The structure of the nucleus is like that of trophozoite. Two types of the cysts differentiated. Small cyst race, their size range from 5 to 10 micron, and large cyst race, their size range from 10 to 20 micron. This size of cysts represents the presence of strains of Entamoeba. So we have no non-pathogenic commensal amoebae, we call it Entamoeba hartmanni. And a virulent minute form, small cyst race, we call it Entamoeba dispar. And virulent magna form, large cyst race, we call it Entamoeba histolytica. So we have three strains, Entamoeba hartmanni, non-pathogenic, and a virulent pathogenic, Entamoeba dispar, and virulent pathogen, we call it Entamoeba histolytica, according to the size of their cyst. So the pathogenic isolate, invasive, known as Entamoeba histolytica, and non-pathogenic, non-invasive, known as Entamoeba dispar. Entamoeba histolytica is observed with ingested red blood cells, erythrophagocytosis, while Entamoeba dispar none. إذن فقط الانتاميبا هيستوليتيكا اللي تحتوي على الانجستد RBCs. Incestation, it is trophozoite will round up, secretion of the cyst wall, hyaline wall, aggregation of ribosome, chromatoid bodies as cigar shape, as we see. It is the black form in the cyst, and two rounds of nuclear division. The one nucleus will produce 4 nuclei, as we see. Trophozoite, immature cyst, and mature cyst have 4 nuclei. The nuclei have the same characteristic feature of the nuclei of trophozoite, with central karyosome and regular chromatoid dot. The fourth stage, it is metacystic trophozoite. The nuclei in the metacyst immediately undergo division to form 8 nuclei, each of which gets surrounded by its own cytoplasm to become 8 small amoebulae or metacystic trophozoite. If excitation takes place in the small intestine. The metacystic trophozoite do not colonize there, but they are carried to the cecum. This is a figure for excitation. It occurs in small intestine. The cyst wall will disruption. The nuclei will divide it from 4 to 8 nuclei. The cytoplasmic will divide it and form 8 amoebiae. The trophozoites migrate to the large intestine. So each cyst will produce 8 trophozoites. So it is incestation for protection and for multiplication here. The life cycle of Entamoeba histolytica. It is passes in one host. It is man. Incubation period range from 4 days to 4 months. Infective forms, it is a mature quadrinucleated cyst. A mode of transmission, we have fecal-orally route. The majority of cases take place through intake contaminated and cooked vegetables and fruits. Insect vector like flies, cockroaches, and rodents act as agent to carry infective cysts to the food and drink. Sometimes drinking water supply contaminated with the infected feces give rise to epidemics, infection. Also, we have oral rectal contact. Sexual transmission by oral rectal contact is also one of the mode of transmission, especially among male homosexuals. This is diagram for the Entamoeba histolytica, the infective stage. It is mature cyst, quadrinucleated cyst, followed after ingestion, followed by excitation, and produce 8 amoebiae, as we said before, which migrate to the large intestine and reproduce by binary fission. Then, under harsh condition, will develop tuberous cyst in the lumen of large intestine. Then, cyst will produce with the stool and evacuated to the environment and will ingested by the second host. Also, life cycle diagram for Entamoeba histolytica. Entamoeba histolytica can cause intestinal infection and extra-intestinal infection to the liver, lung, brain, and the skin. Viability. Trophozoites survive up to 5 hours at 37 degrees and may be alive for 1 hour at 5 centigrade. Cyst stage viable in a stool for 10 days at 22 centigrade. The cyst does not destroyed by chlorination of the water. This is a good note for the Entamoeba histolytica. It is not destroyed by chlorinate the water. The trophozoites absorb food from surrounding tissue, including RBCs, fecal materials, and bacteria, dissolve it by cytolytic enzyme. Entamoeba histolytica live in poor oxygen atmosphere under complete or partial anaerobic condition. It lacks the mitochondria, so it's no need for oxygen in there for their life. Pathogenesis. Entamoeba histolytica, facultative pathogen, normally lives in the human large intestine and feed on the bacterial flora. Approximately 85 to 90% of people infected with Entamoeba histolytica are asymptomatic carriers. Among the symptomatic patients, only 10% will develop severe dysentery or invasive disease. The infection with Entamoeba histolytica implies colonization, the speed of which depends on many factors like general condition of the host, bacterial flora present, the diet of the host, cytotoxic power of the trophozoite upon the leukocyte, and proteolytic enzyme found in the trophozoite, pathogenic activity of the strain, invasive strain or non-invasive amoebae. Pathogenesis of Entamoeba histolytica divided to intestinal amoebiasis and extra-intestinal amoebiasis. For the intestinal amoebiasis, we have symptomatic intestinal amoebiasis caused by invasive strain in which Entamoeba histolytica species, and we have asymptomatic intestinal amoebiasis caused by non-invasive intestinal amoebiasis caused by Entamoeba dispar and some strain of Entamoeba histolytica. For the extra-intestinal amoebiasis can reach to the liver and cause liver amoebiasis, reach to the lung and cause pulmonary amoebiasis, and reach to the brain and cause cerebral amoebiasis. All these type of amoebiasis caused by Entamoeba histolytica species only. For the intestinal amoebiasis, as we said before, we have non-invasive asymptomatic intestinal amoebiasis that's caused by Entamoeba dispar and Entamoeba histolytica. Here, the amoebae colonized on the intestinal mucosa only, not reach further tissue. And we can consider these patients as symptomatic cyst passer and may cause non-dysenteric diarrhea, abdominal cramps, and other GI symptoms. While in invasive symptomatic intestinal amoebiasis that's caused by Entamoeba histolytica only, it causes necrosis of mucosa, lead to ulcer and dysentery. The ulcer will enlargement and lead to severe dysentery, colitis, and peritonitis. The lesion enlarged somewhat as the amoeba reach the muscularis mucosa, then gradually it erodes a passage through the muscularis mucosa to submucosa and then to serosa. They reach to the blood vessel and reach to the surrounding tissue. Here, the amoeba will metastasis and cause extra-intestinal amoebiasis, as we see in this figure. Virulence factor for the Entamoeba, we have surface lectin, which help adhesion of trophozoite to the intestinal cells. And we have killing of the cells with pore-forming peptide. We call it amoeba pore. We have three types, A, B, and C. And we have cysteine proteases, which dissolution of the extracellular matrix. So we have three type of virulence factor. We have surface lectin, amoeba pore 3 type from A to C, and we have cysteine proteases. The three factors of virulence of Entamoeba histolytica affecting by host factors such as stress, malnutrition, alcoholism, corticosteroid therapy, and immune deficiency influence the course of infection. Also, glycoprotein in the colonic mucosa block the attachment of trophozoite to the epithelial cell. Therefore, alteration in the nature and quality of the colonic mucosa may influence the virulence of Entamoeba histolytica. Virulence may also be conditioned by the bacterial flora in the colon. So we have three factors: host factors, glycoproteins in the colon, and bacterial flora. This will affecting the virulence of Entamoeba histolytica. The large intestine parts affected show less or more expanded lesions of varying intensity, ranging from edematous swelling and reddening to pinhead size foci with central necrosis or large and typical bottle shape. We call it flask shape ulcer. So the Entamoeba cause ulcer, we call it a flask-shaped ulcer, which either superficial, does not extend beyond to the muscularis mucosa, or may cause perforation to the intestine. Magnification picture for the ulcer of cause. Also, this is large intestine mucosa caused by Entamoeba. This is histological figure for the flask-shaped ulcer caused by Entamoeba histologica. The trophozoite present at boundary of necrotic and health tissue at the base of the flask, and trophozoite ingested the host cell lead to the dysentery, which means presence of blood and mucus in the fecal material. Deep ulcer, which is limited to the submucosa and or extend deeper to the muscularis layer and cirrhosis, causing peritonitis, pericoacal abscess, and gangrene of the intestine. The chronic intestinal amoebiasis, or we call it a chronic amoebic dysentery, as the lesion become a chronic round cell infiltrating the tissue, then become infiltrated with neutrophil and fibroblasts, which tend to form a wall around the margin of the ulcer. So we have the following changes can observe: the small ulcer involving only the mucosa, and extensive superficial ulcer with hyperemia. Localized thickening of the intestinal wall leading to narrow lumen, formation of the tumor-like mass, we call it amoebic granuloma or amoeboma. So amoeboba is inflammatory thickening of the intestinal wall around the abscess, can be confused with a tumor. This is autopsy for the large intestine and perforated by the infection of Entamoeba histolytica. Asymptomatic intestinal amoebiasis, non-invasive. As we said before, it's caused by Entamoeba dispar and less frequent of Entamoeba histolytica, because we have many reasons make the Entamoeba dispar non-invasive as the Entamoeba histolytica. This is Entamoeba dispar adhere to the host cell in very much same way as Entamoeba histolytica, but it produce only very small amounts of amoebopore A and B and non-potent type C at all. Also, Entamoeba dispar is lacking several genes that code for certain cysteine proteases. The activity of certain proteases in Entamoeba dispar is greatly reduced compared to the Entamoeba histolytica. لهذه الأسباب جعلت الانتاميبا dispar أقل إمراضية وهي غير مخترقة للتشو. Clinical manifestation. This develops as early as 2 to 4 weeks after infection with Entamoeba histolytica, or after a symptomatic period of months or even years. As we said before, we have asymptomatic and symptomatic intestinal infection. In asymptomatic infection, the amoebae will colonize on intestinal mucosa only. We can consider this patient as a symptomatic cyst passer. They pass 1,000 cysts per day, or can cause non-dysenteric diarrhea with abdominal cramps and other GI symptoms. Here, antibodies to Entamoeba histolytica antigens are usually not found in serum. In invasive intestinal form that's caused by Entamoeba histolytica, we have acute and chronic stage. In acute stage, usually begins with abdominal discomfort and episode of diarrhea. The diarrhea of varying duration. The first mishy, then increase mucoid, including blood tangid, so-called red currant jelly stool, or dysentery or bloody diarrhea, in which amoebas can be detected including trophozoids containing erythrocytes. The clinical manifestation of chronic amoebiasis. Here, the ulcer sometimes perforates into the peritoneal cavity, and the symptoms include a recurrent attack of dysentery, constipation, loss of weight, ulcerative colitis, and one or more amoeboma. Amoeboma we defined previously. Obstruction of intestine due to healing processes with scar formation may reduce the intestinal lumen. Table differentiate between amoebic and bacterial dysentery. Bloody diarrhea can cause by Entamoeba histolytica or caused by bacillary bacterial infection. So we have differences between these diseases. I want you to read this table and differentiate between each other. Now we will start with extra-intestinal amoebiasis. This picture indicating that Entamoeba histolytica can metastasize by blood stream and reach to the liver, first primarily to the liver from portal vein, and other sites less frequent like lung, brain, skin, and spleen. Amoeba-free stool common here in this infection, and high antibody titers. Early liver abscess is small oval mass. The decomposing lesion contains a brownish or yellowish pus-like liquid, in most cases bacteriologically sterile. Later becoming a necrotic mass with a grayish matrix of necrosed hepatic cells. Amoebas are often only detectable in the transition zone between the lesion and intact hepatic tissue. Later, the abscess increases in size, the center liquefies, the wall is thick, and the content becomes chocolate reddish mass of hepatic cells, RBCs, and bile fat. This picture indicating the amoebic colitis or amoebic abscess. It is chocolate reddish mass. Amoebic liver abscess, chocolate color, as we see in this picture. Chocolate colored pus, necrotic material, usually bacteria-free lesion, expand and كوليسيز تبدي تلتصق الليجن مع بعض further metastasis to other organs. Pulmonary amoebiasis primarily occur, it is rare. It is دائما تحدث هي secondary to the liver abscess. So primary rare condition occurs even without hepatic amoebiasis. The trophozoites can reach the pulmonary capillaries single or multiple via the portal circulation. Secondary arise as a complication of liver abscess from the liver to the base of right lung, resulting in pneumonia. So, pulmonary amoebiasis, rarely primary. It is rupture of liver amoebiasis through the diaphragm. Bacterial infection is common here, and symptoms is fever and cough. Cerebral amoebiasis, a rare complication of hepatic or pulmonary amoebiasis, or both of them. It is single and small size, located mostly in one of the cerebral hemisphere. Splenic amoebiasis found in associated with hepatic abscess. Cutaneous amoebiasis may develop when the skin is in prolonged contact with the amoeba from any causes, such as areas of draining of liver abscess or colostomy, wound in the site of ruptured appendicular and pericolic abscess. Also, the granulomatous ulceration in perianal region. So, cutaneous amoebiasis, mucosa, but in fluids containing trophozoites. The region infected, it is perianal ulcer, and urogenital. As we see, all extra-intestinal amoebiasis, we can find only trophozoites. There is no cyst in tissue. Immunity. Extracellular protozoa, including Entamoeba histolytica, are eliminated by phagocytosis and complement activation. T helper 2 cytokines are released for antibody production. Antibodies responses includes opsonization, complement activation, and ADCC, antibody-dependent cytotoxicity. Reinfection are possible since sufficient immunity is not conferred in the course of infection. Antibodies are usually detectable in serum in invasive intestinal and extra-intestinal amoebiasis caused by Entamoeba histolytica. Epidemiology, the infection is due to the transmission of mature cysts with the contaminated food like fruits and vegetables, drinking water, or fecally contaminated hands of infected person or carriers. Flies and cockroaches can function as mechanical transmitters by carrying cysts from their feces to foods. The diagnosis of Entamoeba histolytica, we have intestinal amoebiasis. We can diagnose by stool examination. We have micro and macroscopically identification. After iodine or trichome stained slide, we can demonstrate trophozoite and or cyst. Also, we have stool culture and special media. We have mucosal scraping and wet mount stained preparation. We can demonstrate cyst and trophozoite. We have serological diagnosis by indirect heme agglutination test, ELISA, latex agglutination test. And we have molecular diagnosis by PCR by using DNA probe. While extra-intestinal amoebiasis, especially liver abscess, we can diagnose by microscopic examination of pus or aspiration, histopathological examination, and we have serological diagnosis by indirect heme agglutination, ELISA test, latex agglutination test. We have radiological examination like X-ray, ultrasound, CT scan, MRI. and we have stool examination. عندنا ملاحظة مهمة هنا في حالة extra-intestinal amoebiasis we have only trophozoite stage, and in the stool examination we have limited successful diagnosis because we have many stool free of trophozoite in this infection. Diagnosis, as we see, intestinal infection, cyst found in formed stool, cyst and trophozoite in diarrheal stool. We have charcot-Leyden crystals like helminths infection. Serological test for use for chronic and extra-intestinal cases, blood examination for leukocytosis. This is not specific examination. Extra-intestinal amoebiasis, hepatic pulmonary amoebiasis based on identified trophozoite sample only. As we said before, in extra-intestinal infection, we have only trophozoite stage. We have no cyst in the tissue. Treatment for symptomatic intestinal disease or extra-intestinal infection like hepatic abscess, the drug of choice is metronidazole. The control measure of Entamoeba histolytica by using personal and group hygiene, protection of water supply from being contaminated with feces, avoid using human feces as fertilizer before treating it. By the end of this slide, we reach to the end of our lecture. Thank you for your listening.